Thank you so much, Dan. Okay, our first finalist for the James F. Crow Early Career Researcher Award is Matthew Ackerman.
Okay, so just imagine you have some population of organisms that vary for some quantitative traits, such as size, and you're interested in describing the amount of this variation which is due to genetic factors and the amount which is due to environmental factors. So if genetic factors contribute to this phenotype, then you'd of course expect relatives to look similar because they possess similar genes. And this concept is formalized in quantitative genetics in terms of the probability of identity by descent, which the first usage of the term in English was actually from a chapter written by James F. Crow, and that's the quote from the chapter back in 1954. But the basic idea here is that the parents pass on their genes by replication to offspring, and so of course those genes are in the same state in the offspring as they are in the parents, and if they influence the phenotype, you'll see some phenotypic correlation between relatives. And so there's actually 9 different modes of identity by descent which diploid organisms can have, and I've just written the 2 alleles in individual X up on top, X1 and X2, and the 2 alleles in individual Y on bottom, and connected alleles which are identical by descent with those solid lines. So, for instance, with that mode delta 1, all of those alleles have to be identical because they're all identical by descent, whereas in, say, mode delta 9, those alleles are each drawn independently from the population, and so you could just use Hardy-Weinberg equilibrium or whatever to calculate the distribution of states. So once you have these 9 terms, if you're interested in doing some quantitative genetics, you can group these terms in groups that share similar relationships between genotypes and phenotypes. So if you're interested in, say, the effect of inbreeding, you can group the terms where, say, individual X, the 2 alleles in individual X are joined by lines. So that's modes 1 through 4 here, and that'll give you the inbreeding of individual X. Or similarly, if you're interested in additive genetic variation, you can group the terms where there's lines connecting individual X and Y, but you have to do some weighting based on the number of connections in each of these different modes. So, mode delta 1, you have 4 connections. Modes 3, 5, and 7, you have 2, and modes 8, you just have 1. All right, so once you have these 7 different relatedness coefficients, if you've measured the phenotypes of organisms, you have their phenotypic covariance. So you can use linear regression to get some information about the genetic variance in the population for the trait of interest. So you've measured the things in red, you've measured the thing in blue, and you're estimating the terms in green. Now, traditionally, you could measure the terms in red either from a pedigree or with experimental crosses, and that will give you all of these terms. But it's becoming more and more tempting to get these measures just from marker-based studies because of how readily available that information is. Now, unfortunately, for markers, people have only been able to estimate theta xy, or sometimes you can kind of estimate delta under some restrictive assumptions. But there was actually a paper published earlier this year that said it was impossible to estimate delta, which is not the case. I've come up with a composite maximum likelihood method that can estimate all 5 of these terms, and I won't be going into a huge amount of detail. I'll just kind of give a bird's eye view of the method. But you might be wondering, so if you notice the 2 bottom terms here, the additive dominance covariance in inbred individuals and the dominance variance, those are both in inbred individuals, so you might think that in a organism like humans, where there's lots of inbreeding avoidance, these terms might not be important. But I'm going to argue that's almost certainly not the case, because these terms are going to let us say something about the population structure. So even in humans, as we go back in generations, you double the number of ancestors you have each generation. And so before long, you've got far more ancestors than there were unique individuals, and you have to be related to the actual individuals by multiple lines of descent, i.e. there is this inbreeding in your distant past, and you'll need those inbreeding terms to capture that. And this also exposes one of the weaknesses of thinking about relatedness in terms of probability of identity by descent based off of sort of these pedigree methods, in that any pedigree starts at some finite point in the past with individuals you're going to have to assume are unrelated. So we don't have pedigrees going back to the Stone Age. And of course, these individuals aren't unrelated. There can be population structure relating the individuals founding your pedigree, and that's going to influence the genetic relatedness you should be calculating. So we can get around these problems by moving to a concept of relatedness, which is really based on the genetic covariance, which I'll be talking about in a moment. But what we are wondering is we just want relatedness between all pairs of individuals in the modern population, so we don't have to worry about what ancestral populations were. We're just going to define everything based off of what we see in the moment. I'd like to argue that we can think about these things in terms of genetic correlations rather than probability of identity by descent. So this is a sort of Bayesian approach where we're asking how knowledge of one individual's genotype changes our predictions for another individual's genotype. And this will give us, I think, an intuitive definition of what being unrelated means in terms of individuals that are independent conditioning on allele frequencies. And it'll also give us some intuitive interpretations of negative relatedness coefficients that are hard to interpret in terms of probability of identity by descent. So the basic stochastic model that we're using probabilities to analyze, using Bayesian thinking to analyze, we're just keeping our pedigree constant, and we're introducing mutations in a particular historical individual, and the only stochastic process that's going on is basically the allele dropping down the pedigree into the current generation. The sort of first statistics that you can, of course, define are the number of alleles arriving at the current generation at the bottom in red, and which I'll just refer to that count as omega. And so the allele frequency lets us define the prior probabilities that individuals X and Y receive these alleles, although they're not identical to the allele frequency. But for those of you who are not familiar with conditional independence, it's really a pretty simple concept. So if we think about, say, a pair of siblings, X and Y here at the bottom, if we know the genotype of the parents U and V, then knowing the genotype of X doesn't tell us anything about Y, because all that U and V are doing is they're generating eggs and sperm, and knowing what egg V gave to X doesn't tell us anything about what egg it gives to Y. But of course, we're not conditioning on the parental genotypes. Typically, we're conditioning on ancestral allele frequencies. And when you condition on ancestral allele frequencies, X and Y are not independent. So we'll need an additional term there, theta XY in blue, which describes how knowledge of X's genotype changes our prediction of Y. And this should be independent of the allele frequency, so we have to scale it by this term, which depends on the priors. And that term turns out to be the important term to write down for the composite maximum likelihood method, but I won't be giving a lot of details on that particular part. And so I said this also gives us an idea of what's going on with negative correlations. So say if we use genotype, we're learning something about the allele frequency in the population leading to U. But if there's some population subdivision, and drift has been acting independently in the two populations, then if we want to know Z's, the relationship between Z and U might not be very informative because of these two separate populations. And then we'll end up with some negative correlations. So just an example of that. using that same sort of pedigree that's held constant and introducing mutations in fixed individuals and then letting segregation occur randomly. You'll see that if we condition on being polymorphic in that final population, then alleles tend to arrive either on the left subpopulation or the right subpopulation. So that's our negative correlation. Now, this is primarily a methods talk, so I really won't be giving a lot of detail about biological systems, but we have applied this method to a large-scale population genomics project in Daphnia, which is this small freshwater crustacean that lives in ephemeral ponds. And when we look at the whole genome data and analyze for relatedness, we can pick out the sorts of things you'd expect, like genetically identical individuals, which aren't surprising in Daphnia because there is some asexual reproduction. And these relatedness coefficients let us get at some really fine-scale population structure, like, for instance, in the graph on the right. The relatedness between an asexual individual and a sexual individual is typically negative, showing that these are really different subpopulations within the pond that are typically not reproducing with each other. So these methods and some other methods are all implemented in this program, which we like to call MapGD, which stands for Maximum Likelihood Analysis of Population Genomic Data. And you can go online on the Lynch Lab's GitHub website to download the program. And it should have some documentation up there, which I've written to tell you all the different things the program can do, like estimating genotypic likelihoods and allele frequencies. So go ahead and check that out. Although not that many people have used it yet, so there might be a number of bugs you can email me about and get them worked out. So with that, I'd like to thank everybody in the Lynch Lab, all of my friends in the Lynch Lab, in particular, of course, Mike Lynch for encouraging me to do this project, and Tom Doak for encouraging me not to drop out of grad school. And with that, I'll take questions. Yeah.
I hope the projection is okay for the rest of the room. So, I mean, you want to use these coefficients and stuff to get to variance components, right? And
Yes.
genetic architect, and that's what you got at the beginning. So, have you done a comparison in how well it does at that compared to some of the more intuitive approaches?
So we haven't measured the phenotypes in our Daphnia yet, so we don't have a feeling for how well that's working in laboratory populations. Simulations, I've analyzed it some, and it seems to be working well, but I don't compare it to traditional methods because they seem to be undefined in my mind. They're really asking sort of different questions. So for instance, other molecular methods can't estimate most of the terms I'm estimating, so we couldn't do a fair comparison there at all. Or you could maybe define it with a pedigree method, but I haven't looked into how those would compare.